Ladies and gentlemen, thank you for standing by, and welcome to the Nektar Therapeutics fourth quarter 2019 financial results conference. At this time, all participants' lines are in a listen-only mode. After the speaker's presentation, there will be a question-and-answer session. To ask a question during the session, you will need to press star then one on your telephone. Please be advised that today's conference is being recorded. If you require any further assistance, please press star then zero. I would now like to hand the conference over to your speaker today, Ms. Jennifer Reddick, Head of Corporate Affairs. Ma'am, you may begin.
Thank you, Crystal. Good afternoon, everyone, and thank you for joining us today. With us are Howard Robin, our President and CEO, Gil Labrucherie, our COO and CFO, Dr. Jonathan Zalewski, our Head of R&D, and Dr. Wei Lin, our Head of Development. On today's call, we expect to make forward-looking statements regarding our business, including clinical trial results, timing and plans for future clinical trials, timing and plans for future clinical data presentations at medical meetings, the therapeutic potential of our drug candidates, outcomes and plans for health authority regulatory actions and decisions, financial guidance, and certain other statements regarding the future of our business. Because these statements relate to the future, they are subject to inherent uncertainties and risks that are difficult to predict, and many of which are outside of our control. Important risks and uncertainties are set forth in our Form 10-Q that we filed on November 7, 2019, which is available at sec.gov. We undertake no obligation to update any of these statements, whether as a result of new information, future developments, or otherwise. A webcast of this call will be available on the IR page at Nektar's website at nektar.com. With that, I will now hand the call over to our President and CEO, Howard Robin. Howard?
Thank you, Jennifer, and thank you to everyone for joining us on the call today. On today's call, we will provide an update on our pipeline compounds, which include our IO pipeline of Nektar IL-2, IL-15, and TLR agonist candidates, and our immunology program, Nektar 358. We will also review our planned upcoming milestones for these programs and provide our financial guidance for 2020. But before I discuss the advancements we made with our IO and immunology portfolio, I'd like to briefly cover some challenges we faced this year that are outside of the core focus of our pipeline. Starting with Nektar 181, as you know, we made a strategic decision last month to withdraw the NDA for Nektar 181. The Nektar 181 AdCom was the first of several that week that were negative for the opioid class, and it became clear from these discussions that the bar for approval of any opioid compound is much higher than what was established by approvals in the past. Additionally, since that time that we submitted our NDA, the liability in the opioid class has become a significant consideration, with numerous lawsuits filed against opioid manufacturers and distributors. And based upon all of these factors, we made a business decision that further investment could not be justified for a medicine in this class, which would have been at the expense of sacrificing important developmental work for our immuno-oncology pipeline. As we look back at our successful development efforts for this program, I want to thank our team for their hard work, thank the patients and physicians who participated in our clinical trials, some of whom came to speak at the AdCom. We did not take this decision lightly, but believe it is the appropriate action to take as we focus on the advancement of our IO and immunology pipeline. Secondly, as you know, we were conducting the ATTAIN study for our chemotherapy agent Onzield in advanced breast cancer patients who also have brain metastases, which compared Onzield to a chemo agent of choice in these patients. The ATTAIN study was being partially funded from a former partnership we had with Daiichi Sankyo, and you'll recall that the ATTAIN study was designed based upon a doubling of survival that we saw in a subset of patients from the earlier Beacon study of Onzield in advanced breast cancer patients with brain mets, as compared to chemotherapy of physician's choice. The primary analysis of the ATTAIN study was completed late last week, and while Onzield performed at least as well as the physician's choice standard of care for PFS and OS, the study endpoint of improvement in overall survival was not met. As a result, we're planning no further clinical work on Onzield, and we're grateful to the patients and their families who participated in the ATTAIN study. With these actions behind us, our company is highly focused in the core areas of immuno-oncology and immunology, where we believe we have the potential to create transformative medicines for patients. Our IO portfolio is highly differentiated with two strong cytokine programs, IL-2 with Bempeg and IL-15 with Nektar-255, and a small molecule TLR agonist program. This unique portfolio allows us to capture opportunities that span both solid and liquid tumors. In immunology, Nektar-358 is advancing into several clinical studies in multiple autoimmune conditions, the first of which are lupus, atopic dermatitis, and psoriasis, and I'll talk more about those later on the call. Let me first start with Bempeg, our IL-2 pathway program and T-cell stimulator. Earlier this year, we announced a revised collaboration agreement with our partner BMS. Under the new joint development plan, we expanded the BMS Nektar active registrational programs for the doublet of Bempeg and Nivo from the 3 studies that were underway to now include 7 studies in first-line and adjuvant settings across 4 tumor types with more than 3,000 patients. The new registrational program builds upon the opportunity in melanoma, bladder cancer, and renal cell carcinoma, and also adds plans for a phase 2 study in first-line non-small cell lung cancer. In addition to the 3 ongoing registrational trials in first-line metastatic melanoma, first-line cis-ineligible metastatic bladder cancer, and first-line metastatic RCC, we've already launched a new phase 3 study in muscle-invasive bladder cancer, and we are initiating a phase 3 study in adjuvant melanoma. I will let Wei cover the design of these new phase 3 studies in a moment. The economics of the revised agreement reflect BMS's continued commitment to the collaboration. At a high level, if you look at BMS's share of clinical costs for the new joint development plan associated with the 7 studies, it is approximately $1.2 billion. There were also some enhancements to the economics for Nektar, which provide additional and accelerated near-term milestone payments. This includes a $25 million accelerated milestone payment that we received in Q1 of this year with the initiation of the MIBC study. It also includes a new $25 million milestone for Nektar at the start of the adjuvant melanoma study, which we expect will occur in Q3 of this year. In addition, the new agreement includes $75 million accelerated milestone payment at the start of the first phase 3 registrational non-small cell lung cancer study with Nivo. The rest of the economics are unchanged. BMS funds 2/3 of the development cost. Nektar contributes 1/3. Nektar books global revenues. The profit split is 65% Nektar, 35% to BMS. We're also entitled to $650 million in total milestone payments upon the first approvals of Bempeg in U.S., Europe, and Japan, and then $260 million per each of the next 3 approved indications for Bempeg. As many of you know, BMS is currently enrolling patients in our registrational trial in first-line metastatic melanoma, and all of the investigator sites are now up and running. Last year, we obtained an FDA breakthrough therapy designation for Bempeg plus Nivo in patients with metastatic melanoma based on the positive data, including complete response rate from our Pivotal 2 study. The phase 3 study enrolling in this setting has 3 endpoints: ORR, PFS, and OS. The current projected earliest timeline for reaching the follow-up time period needed on the number of patients required for the first interim ORR endpoint is the end of Q4 2020 this year. The PFS endpoint is projected to occur roughly 6 to 7 months later, but as a reminder, this is event-driven and that timing could change. For both ORR and PFS, the results will be analyzed by blinded independent radiology review, so also keep in mind that this process will affect timing for the completion of any data analysis. So the first data readout will most likely be Q1 of 21. As we get closer, we should be able to refine this timeline. As a reminder, ORR is designed as an accelerated approval endpoint. We spent only a small amount of alpha on this, and PFS is the full approval endpoint. With the breakthrough designation, the potential for the doublet in melanoma is quite promising, and as part of our amended agreement with BMS, our 2 companies are excited to expand our development efforts into the adjuvant melanoma setting. This essentially doubles the number of patients that could potentially benefit from this doublet in melanoma and represents a significant opportunity for Bempeg. Given BMS has the leadership position with nivolumab across all lines of therapy in melanoma, we're pleased that Bempeg with nivolumab has the potential to further advance the standard of care in both early and advanced stage melanoma. In bladder cancer, we are enrolling a 200-patient study in first-line cis-ineligible bladder cancer, which is intended to support a potential accelerated approval pathway in this setting. specifically in patients with PD-L1 low expression as defined by a CPS score under 10. We expect the first potential data on the ORR endpoint from this trial in Q2 or Q3 of 21. And to build on this opportunity in bladder cancer, we've also initiated a confirmatory phase 3 study in patients with cis-ineligible muscle-invasive bladder cancer. This gives us the ability to capture the opportunity in both early and late-stage bladder cancer, expanding the potential for BEMPEG and nivo to help even more patients. In metastatic first-line renal cell carcinoma, BMS and Nektar have chosen a comprehensive approach that positions the doublet with a TKI-sparing and a TKI-inclusive regimen. Our phase 3 registrational study evaluating BEMPEG and nivo versus a TKI in first-line RCC is now enrolling nicely, and we are on track to potentially have the first interim OS readout in the first quarter of 2022. The TKI-inclusive regimen development work will start mid-year under the new BMS agreement and is designed to support a registrational path forward in a first-line metastatic RCC study with this triplet. We will conduct a phase 1-2 dose escalation and expansion study to evaluate BEMPEG plus nivo in combination with AXI in first-line RCC to establish the dose and administration schedule for a future registrational trial. Finally, BMS and Nektar agreed on a development path for the doublet in first-line non-small cell lung cancer that we believe positions BEMPEG nicely for a flexible registrational path forward in non-small cell lung cancer. BMS will run a dose optimization and expansion study to identify the appropriate dose regimen, and BMS is paying 100% of the cost of that program. And we will continue our work evaluating pembro with BEMPEG in non-small cell lung cancer in our PROPEL trial, which is currently enrolling patients. This gives us the flexibility in the future to evaluate moving forward with either nivo or pembro in non-small cell lung cancer. We're pleased to have the renewed agreement in place and look forward to this phase of our collaboration. This structure also removes certain exclusivity restrictions from the old agreement for a list of indications for BEMPEG, and so provides us enhanced flexibility to pursue other combinations for BEMPEG. Along those lines, we're exploring the potential of BEMPEG with other checkpoint inhibitors and other mechanisms, and expanding this work is a key role for us this year. In collaboration with Pfizer, we have an ongoing phase 1b/2 study in head and neck cancer and castration-resistant prostate cancer. The study will evaluate BEMPEG and avelumab in head and neck cancer and also evaluate two triplet combinations, BEMPEG plus avelumab plus talazoparib, and BEMPEG and avelumab and enzalutamide in prostate cancer. We're very excited to work with Pfizer on this because of the opportunity in these two solid tumor settings for BEMPEG, particularly in patients with PD-L1 negative tumors. We've also started a study in head and neck cancer in partnership with Vaccibody. The study combines BEMPEG with their personalized vaccine approach and could pave the way for a novel treatment regimen with BEMPEG in this tumor setting. In addition, we have plans to start a study with BioXcel combining their molecule, BEMPEG, and Pfizer's avelumab in pancreatic cancer. As you can see, the BEMPEG program is emerging as one of the largest registrational and development programs in immuno-oncology, and we're excited about the potential of this novel agent to combine with checkpoints and other mechanisms. Turning to our next immuno-oncology candidate, Nektar-262, our TLR7/8 agonist, our phase 1-2 REVEAL study is advancing, and we recently achieved our recommended phase 2 dose of Nektar-262 with BEMPEG. You'll recall that because this was a novel combination, we had to evaluate staged dosing of Nektar-262 with BEMPEG in dose escalation. We've observed high levels of TLR activation in the tumor microenvironment, and the dose escalation allows us to understand PK, PD, and then characterize safety for Nektar-262. Our current plan is to take the recommended phase 2 dose of Nektar-262 into a focused expansion in at least one tumor type, starting with 24 relapsed and refractory melanoma patients. Based upon the biology of the innate and adaptive immune system interaction, we will now evaluate simultaneous dosing of the TLR and BEMPEG to explore the combination's potential in the IO relapsed refractory melanoma setting. The scientific community is beginning to recognize the importance of natural killer cell biology in the treatment of cancer, and as many of you know, this area of research is generating much excitement. So let me now turn to our newest clinical candidate, our IL-15 agonist program known as Nektar-255. Nektar-255 is designed to capture the full biology of the IL-15 pathway to cause both proliferation of NK cells and the expansion of CD8 memory cells, which provides us with a wide range of potential development pathways for Nektar-255. Given the product profile, we're advancing forward on multiple fronts with this program, and Jay-Z will provide more details on the data emerging from this program, but let me provide a high-level overview of the progress on this promising mechanism. First, we're enrolling patients into our first-in-human clinical trial of Nektar-255, which began last year. The study is evaluating Nektar-255 first as a monotherapy, and then in combination with Dara or rituximab in multiple myeloma and non-Hodgkin's lymphoma, respectively. In addition, we have 2 research collaborations ongoing with partners who are entirely funding the research. First, Janssen is conducting preclinical studies of Nektar-255 in combination with a number of their internal oncology mechanisms, and separately, in virology, Gilead is exploring the potential of Nektar-255 in non-human primate studies in combination with a number of antivirals in their portfolio. So Nektar-255 has the potential to have many applications in oncology as well as potentially virology, and we look forward to its progress. Moving on to Nektar-358, our Treg stimulatory program, which is partnered with Lilly. We reported significant progress with this program in 2019. Last year, first-in-human data in healthy volunteers were reported at EULAR, and these data demonstrated the candidate's dose-dependent and selected proliferation of Treg cells. We recently completed the Phase 1b multiple ascending dose study in lupus patients, and we have submitted these data to be presented at this year's EULAR meeting. Our partner Lilly also recently initiated Phase 1b studies in 2 new autoimmune indications of psoriasis and atopic dermatitis, and these studies are ongoing and enrolling patients. Our partner Lilly also has plans to start a Phase 2 dose-ranging study in lupus the middle of this year, and they plan to add another Phase 2 autoimmune indication to the development program this year. So we're very pleased with their commitment and the broad nature of this development program, which reflects the potential of this novel mechanism to treat autoimmune diseases. And with that, I'd like to turn the call over to Wei to review the Phase 3 study designs for BEMPEG. Wei?
Thank you, Howard. I'd like to discuss briefly the comprehensive plan we've developed with our partner BMS for the doublet of BEMPEG plus nivolumab in the melanoma setting, an area where the IL-2 pathway has already been validated. As Howard stated, we have generated breakthrough designation-worthy data in first-line metastatic melanoma from our pivotal 2 study of BEMPEG plus nivo. At a median follow-up of approximately 18 months, 34% of patients had a complete response as determined by blinded, independent central review. 42% had a 100% reduction in target lesions, and 47%, almost half, had a greater than 75% reduction in target lesions. The significance of deep responses in metastatic melanoma and its association with survival have recently been demonstrated by the FDA in a meta-analysis they presented at ASCO 2019. Based on this analysis, patients who achieved a 75% or greater tumor shrinkage in their RECIST target lesions, including patients that achieved a complete response, had a very high likelihood of having the greatest improvement in progression-free survival and overall survival, especially if they were treated with immunotherapy. So depth of response is very highly correlated with survival in melanoma with IO agents. With that context, our data from the pivotal 2 study showing that nearly 1/2 of the melanoma patients had a 75% or greater response reinforces our confidence in the doublet in melanoma. And indeed, as we presented in November at SITC 2019, with approximately 18 months of follow-up, median PFS had not been reached. We plan to share updated data from this cohort at a future meeting in the second half of this year. Our confidence in the potential clinical benefit that BEMPEG plus nivo may offer in melanoma has led us and BMS to initiate a study in the adjuvant setting. This study will evaluate the extended treatment of post-surgical patients with BEMPEG plus nivo, with an endpoint of event-free survival. The treatment duration will be 12 months. We estimate that the study will enroll between 900 and 1,000 patients and will compare the doublet of NPEG plus Nivo to single-agent nivolumab. We are finalizing the protocol with BMS and expect to start this trial in the second half of this year. Due to the long duration of adjuvant melanoma studies, we expect a potential first readout in 2024. With the ongoing phase 3 metastatic melanoma study and the new adjuvant study, BMS and Nektar now have a comprehensive approach to expanding the transformative potential of the BEMPEG-Nivo doublet to more patients with melanoma. In addition, as Howard stated, in January, BMS started the new phase 3 bladder cancer study, which is enrolling patients with muscle-invasive disease in the peri-adjuvant setting. Our ongoing metastatic study in urothelial carcinoma is in the cisplatin-ineligible patients, and this new phase 3 study extends our doublet into earlier disease for essentially the same patient population. In addition, the trial will also serve as the confirmatory study for a potential accelerated approval filing planned with our ongoing metastatic trial. In the muscle-invasive study, we will stratify patients by stage and PD-L1 status. During the neoadjuvant pre-surgical phase, 540 patients will be randomized into 3 arms to receive treatment with either BEMPEG plus Nivo or no treatment at all, which is the current standard care. Then after cystectomy, they will continue on the same pre-surgical treatment regimen for a 12-month period. The primary endpoint will be pathologic complete response and event-free survival. Again, this is a longer study, and our first potential readout is expected to be in 2024. With that, I'll hand the call to JZ to discuss more on our Nektar 255 program.
Thanks, Wei. I'd like to spend a little more time discussing the IL-15 program, Nektar 255, as it is the next large cytokine program in our pipeline that is generating significant interest. Nektar 255 was designed to capture the full biology of the IL-15 pathway, specifically meaning that Nektar 255 is designed to capture all the receptor-ligand interactions available through targeting the IL-15 agonist pathway. As a consequence, Nektar 255 functions as a significant expander of natural killer cells and an agent that promotes the survival and expansion of memory CD8 T cells. The clinical and research community is increasingly recognizing the importance of NK cells and memory cells in the IO cascade. Now, as I just stated, a key differentiating factor for Nektar 255 is that we have engineered it to bind to all forms of the IL-15 receptor, versus other mutein proteins in development that only bind to beta-gamma receptors. We believe that this will translate to enhanced efficacy. For example, we know that in order to support NK cell-mediated cellular killing, you need to induce intracellular granzyme B. And data presented at SITC 2019 showed that we get maximal granzyme B production versus other muteins, and even more than native IL-15. Additional preclinical data we generated to date highlights the various combination opportunities for this candidate. First, we see an opportunity to combine with antibodies such as daratumumab and rituximab that work through an antibody-dependent cellular cytotoxicity, or ADCC, mechanism of action. In the ADCC reaction, antibodies bind to the target cell surface via the antigen recognition portion of the antibody. This coating of a cell with antigen-recognizing antibodies is an immune process called opsonization. These opsonized cells are then recognized by NK cells via the Fc gamma receptors on the NK cells, binding to the Fc region of antibodies on opsonized cells. The clustering of Fc gamma receptors promotes degranulation of the NK cells, leading to killing of the opsonized cells. In this way, the targeted antibody is able to selectively and specifically kill opsonized cells via the action of NK effector cells. However, one of the limitations of the ADCC reaction is that NK cells, the effector cell that actually promotes the killing of opsonized cells, are consumed and themselves depleted in the ADCC reaction, consequently limiting efficacy of the targeted antibody. If we are able to enhance the proliferation and function of NK cells by Nektar-255 and combine that with ADCC antibodies, we could see a very profound effect. In non-clinical studies, Nektar-255 exhibited anti-tumor activity and substantially enhanced in vivo proliferation and activation of NK cells to provide sustained cytotoxic function. In the preclinical lymphoma model, where single-agent daratumumab was ineffective, Nektar-255 treatment in combination with daratumumab increased NK cell numbers and activity in bone marrow tissue and enhanced ADCC-mediated tumor cell clearance in the bone marrow compartment. Now, this is a very important result because it confirmed that Nektar-255 was able to mobilize functional NK cells in the bone marrow compartment, indicating that with Nektar-255, we can generate not only systemic but also tissue-dependent effects. More recently, at ASH, we showed that Nektar-255 enhanced the number and function of both NK and CD8 effector memory T cell populations in the peripheral blood from healthy donors and from patients with multiple myeloma. Nektar-255 was also able to revert the inhibitory status of NK cells for multiple myeloma patients and showed synergy with daratumumab and elotuzumab to significantly increase the status of NK susceptibility of the multiple myeloma cells in a dose-dependent manner. Collectively, these data provide a strong rationale for our first clinical study, which is now underway. The study is evaluating the safety and dose schedule of Nektar-255 as a monotherapy, and then will expand into combination with antibodies that work through an ADCC mechanism, including daratumumab and rituximab. We plan to enroll patients with relapsed or refractory multiple myeloma and non-Hodgkin's lymphoma in this study. The study will also evaluate pharmacokinetic and pharmacodynamic effects as well as anti-tumor activity. We have also introduced a robust biomarker program into this trial to provide a deep assessment of the Nektar-255 mechanism of action. Besides NK cells, we will also evaluate total and subpopulations of CD4 and CD8 memory T cells to study the effect of Nektar-255 on their expansion, activation, and survival. This biomarker-rich early clinical development approach allows us to follow the science in the development and planning for Nektar-255. Our goal is to achieve initial results from the first monotherapy phase of this phase 1 trial this year. In addition, our partners Janssen and Gilead may present data from their respective preclinical efforts with Nektar-255 as well. Now, we also see potential for Nektar-255 in combination with CAR-T and other cell therapies. CAR-T is very effective, but only for a relatively short period of time. By adding IL-15 and promoting proliferation of memory T cells, we may be able to get a much more durable duration of response associated with CAR-T therapy. Our collaboration with Fred Hutchinson has yielded some impressive early data, also presented at ASH. Specifically, researchers demonstrated Nektar-255 prevented tumor growth and increased survival of CAR-T cells when added to a CD19-targeted CAR-T cell regimen in models of B-cell lymphoma. With that update, let me turn the call over to Gil for a review of the financials.
Thank you, JZ, and good afternoon, everyone. This afternoon, we announced our full-year financial results for 2019 in our earnings press release. On this call, I will provide our annual financial guidance for 2020. Starting with our cash position, we exited 2019 with $1.6 billion of cash and investments. With our exceptionally strong cash position, we have decided to repay the $250 million of outstanding senior secured notes on our balance sheet. This will strengthen our balance sheet and improve our annual cash flow, as it will result in approximately $20 million of annual interest savings on a go-forward basis. With respect to cash usage for R&D and operations, we expect to use approximately $350 million in net cash in 2020. This compares to net cash usage of $315 million in 2019. This increase in investment in 2020 is primarily a result of our plans to complete enrollment in the first-line registrational studies in melanoma, bladder, renal cell carcinoma, as well as begin the 2 new phase 3 studies we are initiating this year for the expanded BEMPEG development program under our BMS collaboration. After taking account of our plan to repay the $250 million of debt in Q2, we expect to end 2020 with approximately $1 billion in cash and investments. Now, turning to revenue, our GAAP revenue is expected to be between $140 and $145 million this year. GAAP revenue includes $50 million of new and accelerated milestone payments from BMS under our expanded agreement. The first $25 million of these milestones will be recognized in Q1 for the start of the MIBC study, which occurred in January of this year. And the second $25 million milestone will be in connection with the start of the adjuvant melanoma study, which is currently planned for Q3. Excluding these milestones, we expect the remaining $90 to $95 million of GAAP revenue to be fairly ratable over the 4 quarters of 2020, comprised of the following: $40 to $42 million in cash royalties, $34 to $36 million of non-cash royalty revenue, $11 to $12 million of product sales, and an additional $5 million in other license collaboration revenue outside of BMS. We anticipate 2020 GAAP R&D expense will range between $475 and $500 million, which includes approximately $70 million of non-cash depreciation and stock compensation expense. We expect R&D expense to be fairly ratable over the 4 quarters of this year. In addition to the R&D investment in the new trials in the expanded BMS collaboration, I would like to highlight a few other key areas of focus for us in 2020. In order to meet our planned timeline for BLA filing and potential commercial launch of BEMPEG in 2021, we plan to complete validation of our large-scale commercial manufacturing process and begin manufacture of commercial supplies this year. As a result, BEMPEG manufacturing costs will continue to be a significant component of our R&D expense in 2020. Under our BMS collaboration, BMS shares 35% of BEMPEG manufacturing costs. In addition, we will continue to invest in development of BEMPEG outside of the BMS collaboration, including our PROPEL study with pembrolizumab in non-small cell lung cancer, and in combination with other modalities under our collaborations with Pfizer, BioXcel, and Vaccibody. Our R&D expense also includes the initiation of 2 phase 2 studies for Nektar 358 and the ongoing phase 1b studies in atopic dermatitis and psoriasis. As Howard stated, the first phase 2 study in lupus is planned to begin mid-year, and the second phase 2 study in a new autoimmune disease state will start in the second half of 2020. As a reminder, in our collaboration with Lilly, we are responsible for 25% of these costs. In addition, we will continue to invest in our phase 1/2 work for Nektar 255 and Nektar 262. G&A expense for 2020 is projected to be between $105 and $115 million, which includes approximately $45 million of non-cash depreciation and stock compensation expense. For 2020, GAAP interest income will be approximately $30 to $33 million. With repayment of our senior notes, we expect 2020 full-year interest expense of $7 to $8 million as compared to $21.3 million in 2019. We also expect to recognize between $26 and $28 million in non-cash interest expense related to the legacy Cimzia and Mircera royalty monetization. In Q1 of this year, we plan to record an impairment charge on our income statement of between $45 and $50 million related to the discontinuation of the Nektar 181 program. This impairment charge is composed of two parts: non-cash charges of approximately $20 million and cash payments of $25 to $30 million, primarily related to certain non-cancelable contract manufacturing commitments. As I stated earlier, we plan to end 2020 with approximately $1 billion in cash and investments after repayment of our $250 million in senior secured notes. And with that, we will open the call for questions. Operator?
Thank you. Ladies and gentlemen, if you have a question at this time, please press the star followed by the number 1 key on your touchtone telephone. If your question has been answered or you wish to remove yourself from the queue, please press the pound key. Once again, to ask a question, please press star and then 1. And our first question comes from Chris Shabutani from Cowen. Your line is open.
Thank you. Good afternoon. With the BEMPEG programs in lung, can you give us a sense for how enrollment is progressing, both with the program with Opdivo as well as with Pembro? I think historically there have been some bumps in the road. Can you talk about what initiatives you have put in place that may be helping to engender confidence in your timelines?
Hi, this is Wei Lin. I'll take that question. So regarding our BEMPEG combination in lung, as been stated in the call, our strategy is really two-pronged, in combination with pembrolizumab as well as in combination with nivolumab. First of all, the combination pembrolizumab that's being operationalized by Nektar, and we expect that study has started enrollment, and we expect by the end of the year to have 10 to 20 patient worth of data that has a sufficient follow-up, at least 2 scans, to allow us for an assessment of activity. The combination with nivolumab, that's being operationalized fully by BMS, and that study has not opened yet, and then we'll provide more details as the year goes along.
Great. And then with 255, which seemed to be an asset that Jay-Z highlighted here, can you give us a sense, maybe frame what kind of efficacy results we may see in the monotherapy setting for those 2 indications that we are likely to see data, the myeloma, et cetera? Thanks.
Sure. Hey, Chris. This is Jay-Z. We'll be exploring patients that have pretty late-stage disease, because remember, this is a first-in-human study. So we're dose escalating in a typical way with small numbers of patients at any given dose level. And we're treating patients with very advanced late-line disease, kind of like in a salvage regimen. So we'll be monitoring all of the possible outcomes that you can get in a study of this kind in a first-in-human setting. In addition to any responses that we may see in these liquid tumor settings, Chris will also be deeply diving into the mechanism of action of IL-15 pathway agonism. So we'll be looking for cellular changes systemically. We'll also be looking, in the tissue, for example, like in the bone marrow compartment, where we expect there to be additional cellular changes induced by this mechanism of action. So we'll be looking to present the totality of the results as we can this year.
And remind me in what forum or setting that data may come, at a medical meeting, or will you be top-lining? Will it be ASH?
Yeah, it would most likely be at a medical meeting. That's correct. So that's a good example. There are a couple of others that you could think of where liquid tumors are the focus.
Sounds good. We'll look forward to that data on 255 program. Thank you.
Thank you. Our next question comes from Alexander Duncan from Piper Sandler. Your line is open.
Hi, thanks for the questions. For the 255 program, have you had discussions to evaluate 255 in bladder cancer as a doublet or triple, given some positive IL-15 superagonist data there recently, and you're doubling down in bladder with BEMPEG? And then maybe more broadly as a platform, what gives you the most confidence that Nektar's compounds will be the ideal cytokine mimetics to combine with moving forward as we see more and more competitors entering the cytokine space? Thanks.
Yeah, so there were 2 questions there. Let's start with the first one. So in the setting of Nektar 255, we like you stated, we appreciate that this is a very broad mechanism. and it shares a lot of features that are non-overlapping with an IL-2 kind of pathway engagement. And you can apply that IL-15 pathway agonism broadly, not only in liquid tumors, which is the first in human study, but also into the solid tumor settings. And there are a number of tumors that are characterized with NK cell infiltrates, and bladder cancer is one of those. There are others that share some features of the GU tumor kinds of settings as well. So there are definitely very, very broad application opportunities. And it doesn't even just stop with 255, because it also goes with the combination partners that are selected. For example, when you consider targeted therapy as the combination partner, you know that you have an incredibly wide range of targeted therapies across many, many tumor types, including large indications ranging from things like breast cancer with agents like Herceptin to all of the rituximab, B-cell, NHL kinds of opportunities. So, yeah, we'll be looking for a very broad set of development opportunities for 255. And on the second question about the cytokines that Nektar works on, I think that we've sort of had the chance to really revitalize and inject a whole new momentum into the cytokine field with the results that we were able to deliver with BEMPEG. And obviously, we've been building off of that in the sense that we've been advancing the BEMPEG program very aggressively and very, very quickly into multiple registrational settings. We've kind of set a bar and a hurdle for other companies that are following behind us. We obviously have a deep belief in our approach to targeting cytokines, and our molecules have differentiating properties that we built into them with our polymer chemistry conjugation platform. And those other companies that are behind us, they have to chase us in that regard.
Awesome. Thank you.
Thank you. Our next question comes from Jessica Fye from JP Morgan. Your line is open.
Hey guys, good evening. Thanks for taking my question. Can you talk about whether you might pursue the potential to shift the interim in the first-line phase 3 melanoma trial to one based on complete response rate? Would the decision to change it be based on feedback from the FDA that success on CR could serve as a basis for accelerated approval? And if so, have they given you any feedback on that front?
Hi, this is Wei Lin. We certainly, to a large extent, the breakthrough designation we obtained in first-line melanoma was large to a large extent based on the based on the complete response that was seen in our pivotal 2 cohort. And that it was very encouraging in using that endpoint in assessing the quality and totality of our data. Now, we do have ongoing discussion with agencies, multiple agencies worldwide, regarding that endpoint as the first endpoint to take a look at, and that's something that we'll provide further update as we complete those regulatory discussions. So right now, the formal first primary readout is still based on ORR.
Great, thank you.
Thank you. Our next question comes from Bert Hazlett from BTIG. Your line is open.
Yes, thank you. Thank you for taking the questions. I have 2 of them. First is, it seems that you would, in the phase 3 in first-line melanoma, you would want to match the patient characteristics of Pivotal 2. Can you give us any anecdotes that you've been able to do that? Again, phase 3, the pivotal study versus the Pivotal 2 results that you've shown. And then secondly, Jay-Z, you mentioned 262 and the safety. Could you want to share any characteristics of the safety of that program you saw in phase 1? Thank you.
Hey, Bert, this is Jay-Z. So one of the things that we did in the Pivotal 2 cohort is we made sure that we enrolled patients that had the same array of baseline demographics and risk factors that would match the phase 3 patient population. So, for example, about a third of our patients presented with liver metastases that entered into the Pivotal 2 cohort. Also, about a third of our patients presented with elevated LDH, lactate dehydrogenase levels above the upper limit of normal, as well as being stratified into the M1C and M1B, which are the more sort of severe or worse prognostic factors for patients with melanoma. And the reason why we did that was we ensured that that was a patient population that would match our phase 3. And in fact, if you look at the results that BMS published for Checkmate 067, the proportion of patients that had liver mets or elevated LDH or M1C status is actually very, very similar to what we have in our PIVOT-02 cohort. So that's a very important point. I'm very happy you asked that question. And for us, it was very important because when we looked at the overall PIVOT-02 cohort data for melanoma, not only was it important that we presented it by a blinded independent central review, right, to increase the strength of a single-arm cohort, but also that the patients within that cohort had many poor prognostic factors. And, we presented in that last update at SITC that even patients with liver mets had complete responses, right? So even the most severe case, the most severe setting, there was still a very, very deep response observed, and it's a patient population that matches the phase 3 population. So that's an important point. And then to your second question, you were asking about the safety with 262. So definitely one of the things that we did in developing 262 was create an intratumoral administration, route of administration strategy. And we used our polymer conjugation to enrich the residence time of the agent in the injected tumor. And all that is done to minimize systemic exposure. And the good thing is that because this is a TLR7/8, it's based on, a pathway that we really have a lot of experience in clinically. We're able to compare the results that we're seeing in our study relative to the results that you'd expect for systemic infusion of a TLR7/8 agonist. And, as we presented preclinically, and as we saw in our toxicology studies and others, intratumoral injection is a very, very different and a much better tolerated way to administer a TLR agonist. Certainly much, much better than giving it systemically where you have, massive kind of systemic cytokine effects. In the intratumoral setting, it's all very localized, and you really don't see that.
Terrific. Thanks for the color. Look forward to the data.
Sure. Thanks, Bert.
Thank you. Our next question comes from Paul Choi from Goldman Sachs. Your line is open.
Hi, good afternoon, everyone, and thanks for taking our questions. I have two. The first is on the phase 3 metastatic melanoma program with regard to timelines. I think the trial is still indicated on ct.gov to be enrolling patients, but as you're thinking about an interim readout in the fourth quarter, given the fact that we saw with the PIVOT-02 that the time to response for complete responses was about 8 months. Can you maybe just comment on how we should, sort of frame what kind of CR rates we should see, just given what might be sort of a shorter treatment duration or exposure to the drug during the time period ahead of it? Thanks.
Yeah. Thanks for the question, Paul. So it's been stated earlier. I think the endpoint has not been changed to this not CR yet, and CR, as you pointed out, the median time to CR is about 7 months. And because of that, if we were to do a CR analysis, we would like to follow the patient for probably a 10-month duration. And so there's going to be ongoing discussion with the health authorities regarding the sample size and number that would be required for registration based on the CR endpoint. And based on that, followed with a sufficient duration, that's going to determine the readout of the CR if that were the endpoint. So some of that still is yet to be decided. I think by our current estimate, it's probably going to be roughly around the same time as the current projected ORR, but obviously this is all based on current projection and in absence of the full health authority interactions. So those things will be made clear in the coming month. Thanks.
Okay, thanks for that, Wei. And then just on 358, as you and your partner Lilly explore the advancing the program in the various dermatological indications, these are sort of fairly competitive areas, and I guess just as you think about efficacy bars, whether it's in AD or other indications, are you with the next set of data updates, are you looking to for any particular metrics or any points of particular differentiation? Any color there you could provide would be helpful. Thanks for taking our questions.
Sure. Hey, Paul. Yeah, so this was a very similar question you asked a couple of days ago. So in the setting of the dermatological, uses, definitely the benchmarks are out there, right? And we know like agents like IL-23 and others they're setting some very unique bars in those settings. So we're aware of those. Lilly is aware of those, and so those are important benchmarks for, judging all of the nature of advancement in dermatological indications. So that's always there. The second thing that is, though, really important is that this is a novel mechanism that's being applied into this dermatological setting. And so, in fact, we know that there's even expectations of Tregs moving within to the skin where you have areas of dysfunctional dermal, cell growth. For example, we'd expect a vulva in psoriatic patients to be infiltrated by Tregs, or an AD plaque to be also infiltrated by Tregs. So we'll be looking also for that kind of mechanism of action because that adds to that whole assessment. And then the other is, remember that in these kinds of diseases, you see themes of kind of similar autoimmune dysfunction. The tissue is different because tissues represent different kinds of inflammation. But in terms of the immunology, we see similarities. And in that regard, an indication like atopic dermatitis is very important because there are many indications that have atopy as an underlying disease pathology driver, not even beyond atopic dermatitis. That's just atopy in the skin, but we can also see atopy and allergy in many other kinds of settings, right, beyond the dermal compartments. So those are very important studies that give us both a sense of opportunity within those specific derm indications and also how to even more widely consider and broadening the Treg mechanism into the additional autoimmune themes covered by those two indications.
Thanks for the color, JD.
Sure.
Thank you. Our next question comes from George Farmer from BMO Capital Markets. Your line is open.
Hi, thanks for taking my questions. I'd like to ask more about using complete response as an endpoint in the phase 3 melanoma study. Firstly, is Bristol Myers on board with this approach, number one? And number two, how do you think about using CR as an endpoint? What is the delta that needs to be achieved, do you think, in order to determine whether the trial should be submitted for an accelerated approval?
Yeah, so first of all, we certainly, BMS is a partner with us. On this, we have had extensive discussion, and based on the enthusiasm FDA has shown with the CR endpoint, BMS is also very much in agreement that this is a valid endpoint that we should really pursue. And so now in terms of the bar, so the bar is nivolumab, which is a control arm in our ongoing studies, about 8 to 9%, with sufficient follow-up that we expect to be at the time of the readout. So that would be the benchmark, and we'll probably target something around 15% improvement would be adequate to statistically exclude the possibility of this being a type 1 error. So that's so right now, just to remind you, based on our pivotal 2 data, our CR rate with about 18 months of follow-up is about 34%. And in fact, even back with a 12-month follow-up, it was at 34%, right? So I think that's what we're trying to project based on that number. So at least if we're able to replicate what we've seen in pivotal 2, we have a good degree of confidence that we can clear the bar of improving upon this past CR being seen in nivolumab. Thanks.
Okay, yeah, that's very helpful. And then, do you think when you ultimately do present more pivotal 2 data, do you think we'll be able to see some overall survival results from that study?
Yeah. So in pivotal 2, the melanoma cohort, we're continuing to follow. There are still a number of patients that are on treatment, and there's at least 2 patients who have potential of converting from PR to CR. And in second half of the year, we would like to provide update on potentially two changes. One is deepening response and any potential patient changing from PR to CR. as well as a new estimate on what the possibility of PFS would be. As a reminder, with 18 months of follow-up, we have not identified the median progression-free survival yet, and with longer follow-up, we can estimate even a longer PFS potential. And as a reminder, nivolumab really has about 7 months of PFS, 6 to 7 months of PFS. So that is the benchmark in our current study.
Great. And then finally, on Nektar 358, the fact that Lilly is moving forward into a phase 2 in lupus, does that imply that the phase 1b data is looking good? I mean, what drove that decision to move forward?
Hey, George, this is JZ. So with Lilly, remember, as we entered into an agreement, they'll be running at least four actual phase 2 studies across multiple autoimmune indications. We certainly ran the phase 1b. Nektar completed that study. That was a multiple ascending dose study in patients with mild to moderate lupus. And then this phase 2 study obviously is now diving deeper into the lupus population. Obviously, it'll be treating them for much, much longer duration than a multiple ascending dose study does, and also moving into more moderate to severe disease for, therapeutic benefit. Now, why lupus, right, is a great question that you asked. And actually, lupus has a very deep biomarker that's driven by regulatory T cells and then the application of Nektar 358. Lupus patients have a well-known and well-characterized dysfunction in their regulatory T cell compartment. And we see this manifested in two ways in patients with disease, especially progressive disease. We see both a lowering in the number of Tregs in circulation relative to healthy patients that don't have lupus. And you also see a decreased suppressive capacity. So when you actually isolate Tregs from lupus patients and you test them in a functional assay, they're very, very poor at immunoregulatory function relative to Tregs, isolated from healthy donors. So both of those represent two key sort of therapeutic themes, right, for applying Nektar 358 and this mechanism of action into this disease. So we're looking forward also to presenting some data from the phase 1b study in lupus patients later this year. We already submitted an abstract to EULAR to present that data. We look forward to giving an update on that from the phase 1b, and we look even more forward to Lilly starting their phase 2 study later this year.
Great. Thanks, JZ.
Thank you. Our next question comes from Jay Olson from Oppenheimer. Your line is open.
Oh, hey, thanks for taking the questions. Can you talk about the data that you have supporting your decision to move the doublet into muscle-invasive bladder cancer? And do you expect to see efficacy in all comers, or will it depend on PD-L1 expression level or tumor mutational burden or some other factor?
Yeah, this is Wei. Thanks for that question. So the data to support us going to muscle-invasive but bladder cancer is the same data set that support us going to metastatic disease. And there, in fact, in the data we have shared publicly, we've actually seen benefit in the PD-L1 negative as well as the positive. And so it is all comers in the sense it could be regardless of PD-L1 status, but it is limited to the cisplatin eligible population, just like in our metastatic study.
Okay, great. And then in RCC, can you describe the rationale for pursuing a TKI sparing regimen? Is that based on a potentially superior AE profile, or are you looking for superior efficacy?
Yeah, it is certainly based on superior AE profile. As you know, that the level of grade 3 AE, whether you look at avelumab plus axitinib or pembro plus axitinib, is actually very, very high. It's actually over half the patients experience grade 3 or higher AE during the study treatment. And there's discontinuation due to that. And so to really provide maximum benefit, we expect if we can keep the patient on drug, that's the way to really provide the maximum benefit. And in addition, by using both the TKI as well as the checkpoint in one regimen as a first line, you basically exhaust most of the available therapy. And if we can provide a TKI sparing regimen as first line, the physician and patient still having reserved a TKI option as second line with subsequent therapy. I think all those are benefits to the patients. So those are multiple considerations that our initial study that really has been designed via TKI sparing. Obviously, there are some patients who are not eligible for TKI due to a variety of reasons. And so we thought it very important to provide that option to patients for all these reasons. Thanks.
That's super helpful. Thank you very much for taking the questions.
Thank you. Our next question comes from Arlinda Lee from Canaccord. Your line is open.
Hi, guys. Thanks for taking my questions. I was interested in IL-15. There seems to be a number of other competitors in this space roughly entering the clinic right around now. I was curious, what do you think are some key differentiators and early reads that we should look at to maybe help assess or assess the competitive landscape? Thank you.
Sure. Hey, Arlinda, this is JZ. So certainly, like, one of the things about IL-15 is it like other IL-2 family cytokines, like IL-2, 15, and 7, all the ones that are the same class, they have multiple receptor ligand interactions as part of their biology. So there's usually a dedicated alpha receptor, right? And then there's a common or shared set of beta gamma receptors. And of course, gamma, as you know, is common gamma chain that's shared with like over 50 different cytokines that signal through that. And so the receptor ligand interactions, the cells that express the different receptor complexes and all the modes of binding, we believe are very important. And in particular for IL-15, there's at least 3 modes of binding. So there's IL-15 alpha dependent and independent binding. And if it's independent, then it works kind of like a soluble ligand interacting with the IL-2 receptors beta and gamma, functioning kind of sometimes called as a superagonist. And then there's 2 different modes of IL-15 alpha dependent binding. One is when it's on the same cell, we call that in cis, or on a different cell, we call that in trans. And in the trans binding, it's particularly important because that's driven by cell-cell contact. And a lot of IL-15 signaling is driven at the cell-cell contact level when, one cell presents on an alpha-bound IL-15 molecule onto a neighboring cell that has the beta-gamma receptor complexes. Now, our molecule, Nektar-255, is very different from all of the competing pipelines because in all of those settings, they have IL-15 in complex with the IL-15 receptor alpha. So really, like the Altor molecule or the Xencor and others, or the Novartis' Sushi domain molecule, they all really share just one primary mode of binding, and that they only interact with the IL-2 receptors beta-gamma. They're unable to interact in either the cis or trans alpha-dependent binding modes. So whereas Nektar-255 preserves all of the available receptor ligand complexes, and we designed it that way on purpose. So firstly, that's a scientific differentiation at the receptor level. And then we've shown examples of what that means biologically. So we can induce a higher granzyme induction. We can induce interleukin-18 expression in CD14 monocytes, and we've presented that data publicly. And those are 2 activities that the receptor complexed versions of IL-15 targeting were unable to sustain. The other thing that I think is really important to look at is if you see evidence of kind of tachyphylaxis. With repeated dosing, is there a loss in any biological effect? And that's going to be another one really important place to look at, because I think that's going to be a place where a molecule like Nektar-255 will really distinguish itself, as it should have far, far less or no evidence of tachyphylaxis relative to some of the other pipelines.
Is the tachyphylaxis something that might show up in phase 1?
Yeah. So there's been publications from other molecules. For example, there's a publication from the Altor molecule that shows in the clinic that upon repeated dosing, there's a loss of biological effect, specifically at the level of cellular expansions they have measured in the blood of patients treated with that molecule.
Thank you.
Sure.
Thank you. And our next question comes from Andy Shea from William Blair. Your line is open.
Great. Thanks for taking my question. So in RCC, it's kind of interesting because in the frontline setting, I think CR drives adoption. And if I remember correctly, the CR rate within the RCC cohort was about 4%. So just curious if, I guess last year I think the company had planned on presenting data at ESMO. Just curious about the next steps of data release for that cohort.
We have shared data in the last earnings call towards the second half of last year. So there's been no new update regarding the RCC data. So we don't have any plans to have another presentation in a major medical congress on the RCC data, but we do eventually plan to present that in a manuscript form.
Got it. Okay. And just kind of follow up on Bert's question on 262. So is the strategic positioning here that you'll have a potential combination 262 and BEMPEG as a salvage, as a checkpointless salvage regimen in various solid tumors? Is that a correct assertion?
Hey, this is Jay-Z. So remember when we presented this program for the first time, we described the rationale for 262. So in providing an innate signal to BEMPEG, which is an adaptive signal, right, you have a comprehensive kind of an immune response. And we developed it in addition so that it's really a wholly owned doublet that Nektar has. And really it was developed specifically and expressly to be used with BEMPEG. So then in terms of all of the uses of it, it could be quite broad. We're choosing to begin the clinical development in evaluating the activity in patients with very late stage disease. So as you know, REVEAL is kind of like a basket study with very advanced patients with solid tumors, rolling multiple different tumor types. But as we mentioned, we're going to focus on certain tumor types, such as melanoma in the relapse refractory setting as we move forward. And you know that we'll be evaluating the doublet as well as a triplet by adding a checkpoint inhibitor as well to the combination of Nektar 262 plus BEMPEG. Now, there's really a lot of opportunity to use this kind of a combination. We'll be beginning, but like I said, we're beginning the development in the relapse refractory setting. One of the things that we saw preclinically with the program, remember, was a really deep set of abscopal immunological responses. And we presented data showing, for example, animals that have multiple tumors. But you only needed to inject one tumor and only one time in order to get the full benefit of the immune reaction moving throughout the animal's body and really eradicating any tumor, no matter where it was, no matter how distal it was from the original injected tumor. And this is a response that we call in cancer immunotherapy an abscopal kind of an immune response. And so, again, that was really one of the predicated visions of the program. And so as we move into that setting and as we demonstrate that the molecule can work in that way, that could give a lot of very broad use and broad opportunity for that regimen.
Okay. And in terms of dosing in non-small cell lung cancer, so, you know, the increase in dosing relative to other pivotal studies, is that a non-small cell lung cancer specific phenomenon, or how do you think about just kind of cross-indication differences in dosing?
Yeah, I think there were a couple of thoughts that underlie our plan to both optimize our dose as well as expand into non-small cell lung cancer. Certainly, we stand by our 6 microgram per kilogram dose, which is in all the registration studies in melanoma, bladder cancer, and RCC. And that's also supported by the pivotal 2 data that enabled and gated the registration trial with our partner BMS. And certainly in melanoma, the data is sufficient for a breakthrough designation. So the clinical activity is clearly there. I think as we take a look at the treatment landscape for non-small cell lung cancer and also the biology disease, and the fact that monotherapy requires a biomarker all this highlight that the challenge of non-small cell lung cancer and makes us think whether a different dose could provide a greater benefit there. That's one. And the other is, since we treated around 500 patients or so with a combination nivolumab plus BEMPEG, we've learned a lot about the safety profile of giving BEMPEG together with nivolumab. And from all those lessons, we were able to control the AE quite well, much better than when we started the program. And we believe with the current management guideline, we can actually dose BEMPEG at a higher dose, and that's another hypothesis we certainly like to try. And that's another reason why we're undertaking the dose optimization. So if we're able to achieve a higher dose, then potentially in the long registration study, we'd be using that dose to move forward.
Okay, and last question for Gil. Just kind of remind us in terms of the R&D reimbursement from Bristol. I think there's a cap, there's an annual cap, and the access amount would basically get reimbursed in the first quarter. So how do you, from a modeling standpoint, how do you kind of anticipate that as we head into the first quarter?
So you're right, Andy. There remains a $125 million a year cap on R&D expense sharing under the agreement, and that's under GAAP revenue. And we expect this year that we will exceed the cap towards the end of the year. And the way the agreement works is that we would be reimbursed for exceeding the cap in the following quarter. So that cash would come in 2021.
Okay, great. Thanks for answering all my questions.
Thank you. And again, ladies and gentlemen, to ask a question, please press star and then 1 now. And we're going to take our last question from Dana Graybosch from SVB Leerink. Your line is open.
Thank you so much for the question. The first is a clarification on the early stage melanoma trial. Is this going to be an adjuvant trial or a neoadjuvant adjuvant trial?
The registration study is an adjuvant trial. It's conformed with the currently approved label for nivolumab, which will serve as a control arm for the study.
And a follow-up on that, because in melanoma we've seen really dramatic benefit in the neoadjuvant setting with IO, especially the combination, but also single agent. Do you worry about missing out on that benefit, or will the patients be getting nivo alone in the neoadjuvant setting here?
That's certainly a possibility. I think current standard practice, that's actually not the standard care. There's no approval in the neoadjuvant setting because most of this data are generated in very small cohort of patients, really in the teens. And so I think for an approval in the neoadjuvant setting, there has to be a full randomized trial as what's been done for adjuvant study. And certainly right now there's no undertaking attempt by any of the sponsors to undertake such a large study, given the standard care is already established in the adjuvant setting. So now some academic centers can practice, and some physicians may treat themselves, may treat their patients using a neoadjuvant approach for selected patients. But I think the predominant clinical practice is not going to be neoadjuvant, and hence we don't expect in the foreseeable future, certainly within the time frame of our study reading out, the neoadjuvant is going to overtake adjuvant as a standard care. Thanks.
Thank you for that. Second question back on the CR as a potential endpoint in the frontline metastatic melanoma study. It's interesting because usually response rate is very quick, so you get a snapshot of a certain amount of follow-up. But I think we all know that in melanoma, if you look at the 3 or 4 year CR rate, it continues to deepen with nivolumab or pembrolizumab alone. And I wonder how you're thinking about that, whether you'd have to have this data mature over time, getting an interim, but also looking at the comparison of CR rate over a number of years.
Yeah. So, certainly our confidence in this potential use of CR as a basis for accelerated approval is based on our pivotal 2 data. As been pointed out previously, our median time to CR is actually 7 months. So it's actually fairly fast. I understand there's based on Checkmate 67, there's very long follow-up, up to 5 years. And it is true that with prolonged follow-up, the CR does steepen over time. But even if you look at the nivolumab or nivo-ipi data, with prolonged follow-up, even after 4 to 5 years of follow-up, nivo has not achieved a CR higher than 20%. And nivo plus ipi has barely achieved a CR rate of 21%, which is very different than what we've able to accomplish just with a little over a year follow-up of 34%. So that is really the distinguishing factor, right? So at the rate of CR that the BEMPEG plus nivolumab has been able to achieve in the pivotal 2 study is much quicker. And I think during the time of follow-up, even with what we plan to have a 10-month of follow-up, we think we can really truly distinguish ourselves from the nivolumab control arm.
Great. Thanks. And one last question for JZ around Nektar 255. We know a lot of patients, especially hematological malignancy patients, have exhausted or dysfunctional NK cells. And I'm wondering if Nektar 255 can restore their function and proliferation as well.
Yeah, that's definitely one of the things that we're very, very excited to look at. And it's part of the reason why the starting patient population that we have in our study are late-stage patients with NHL, and in particular late-stage patients with multiple myeloma. You know Dana really well, right? Particularly in multiple myeloma, there's a massive and can be catastrophic dysfunction in the NK cell compartment, both in terms of cells that have poor effector status that can't maintain, ability to degranulate, and cell numbers that go down. Even after agents like DARA, for example, or any CD38, that depletes these cells further as well. So definitely that's one of the earliest biomarkers that we're looking at in our clinical trial. As you know, with these cytokines, we measure systemic cell changes all the time. And because it's a blood-based central compartment measurement, it's very robust and easy to measure. So it's definitely a key part of our biomarker program and our mechanism of action testing for Nektar 255.
Great. Thank you.
Thank you. And that does conclude our question and answer session for today's conference. I'd now like to turn the conference back over to Mr. Howard Robin for any closing remarks.
Okay, well, thank you everyone for joining us today. And as usual, I'd like to thank our employees for their hard work and their commitment to our company. Nektar has really built a highly valuable pipeline of programs in immuno-oncology and immunology, and we've really tried to address large patient populations where large numbers of patients are suffering from serious disease and have a high unmet medical need. In solid and liquid tumors across many potential treatment indications, and in autoimmune disease across multiple autoimmune and chronic inflammatory disease states. So we look forward to advancing these programs, continue to provide you with progress on our growth, and we look forward to seeing many of you at conferences over the next several months. So thank you very much for joining us today, and we look forward to seeing you. Thank you.
Ladies and gentlemen, thank you for participating in today's conference. This does conclude the program. You may all disconnect. Everyone, have a wonderful day.